The last pattern of inheritance that we're going to discuss is an optional one, really interesting one, and pretty rare, actually. If you think about how many genes we have in the genome, we got about 20,000 genes. Only about 100 exhibit this pattern of inheritance that we're going to discuss, which is genomic imprinting. In this phenomenon, what we see is that we don't have expression of a gene from both of the homologs in the cell, right? So the gene is only going to be expressed from either the allele that was inherited from mom or the allele that was inherited from dad. So we see monoallelic expression of the gene, right? And the way that this comes about is because of an imprint that gets applied to the silenced gene, right? So there's a silencing imprint, a mark, a molecular mark, and usually this is in the form of DNA methylation in a region of the gene that regulates its expression, right? So DNA methylation leads to silencing of the gene. And so whichever allele is methylated, that's the one that is not expressed, and the allele that is not methylated is the one that is expressed. Now, when does this happen? Well, this actually happens in the gonad during gamete production, during meiosis. All of the methylation marks across the genome are erased and reset to match the new parent of origin. So if a gene is always methylated on the maternal allele, then in females, when they make their gametes, they're going to be methylating that specific gene in every single gamete that they produce. Whereas in males that are having ongoing spermatogenesis, that gene would not be methylated. So this happens during gamete production, and then that methylation pattern, once established, is maintained in the offspring that are produced by fertilization, the combination between those gametes. So that individual that's formed is always going to see that allele of that gene remain methylated across cell divisions. The mark is maintained and reestablished after every cell division, and that allele is always going to be silenced throughout their life. So as I said, there are about 100 genes that exhibit this sort of a phenomenon with inheritance. This is a form of epigenetic inheritance because it's influencing the expression of the genetic code. Epigenetic rather than strictly genetic, because it's not actually altering the DNA sequence. We're not talking about mutations here, but we're talking about covalent modification of the DNA nucleotides themselves that's changing the way the genetic code is being expressed. All right. Now, if we think about it, it seems like this should be a dramatic disadvantage, because essentially what we're doing is we're making ourselves pseudohaploid for those 100 or so genes that we've got in the genome that are being expressed this way. And there's a lot of discussion and a lot of controversy right now trying to come up with explanations for why we do this and why genes are methylated. And there are a lot of competing hypotheses that I'm not going to go into in the video. If you're interested, I can point you towards some resources and maybe a extra credit opportunity or two to investigate that more fully. But as I said, it's a pretty rare phenomenon, but it does have clinically relevant impacts, because there are mutations in some of these genes that are imprinted, or deletions or chromosomal abnormalities that involve these imprinted genes, and that can have very significant clinical outcomes, as we'll see. So when we have these sorts of imprints established where only one allele, the maternal or the paternal allele, is being expressed, this is going to lead to parent of origin effects, where an individual's expression of a mutation that they've inherited, for example, is going to depend on which parent they inherited the mutation from. So to see how this might play out, let's think about an example from mouse. So all placental mammals have imprinted genes. It's not always the same imprinted genes across different species. But if we look in mouse, we can see that they, like us, have about 100 genes that they express in this imprinted fashion, one of which is a gene called insulin-like growth factor 2, or IGF-2. This gene is imprinted in such a way that the paternal chromosome, so the chromosome that an individual inherits from their dad, expresses the IGF2 allele, whereas the maternal chromosome, the chromosome that they inherited from their mom, is marked with a methylation imprint, right? This little asterisk is supposed to signify methylation, and silenced. All right. So if we think about the normal scenario in a mouse, if we have normal IGF2 alleles, so no mutations present, normal IGF2 alleles, and the normal imprinting pattern in every cell of this mouse, the allele, the homolog containing the allele that was inherited from mom is going to show methylation at the IGF2 gene, only on the maternal chromosome. And so only the IGF2 gene on the paternal chromosome, the paternally inherited chromosome, is going to be expressed. So this mouse is expressing half the amount of IGF2 that it would otherwise because there's no expression coming from this maternal allele, right? So that's the normal scenario, and that's what we see in the wild type population, and that leads to normal sized mice. Now this is a growth factor. IGF2 is a growth factor, and if we disrupt its expression, either overproducing it or underproducing it, or hyperactivating it or inactivating it, then we're going to see effects on growth size. And so there are mutations that have been identified in this gene that knock out the function of IGF2 and prevent its expression. And so if we have a decrease in IGF2 expression, then that results in a phenotype of a dwarf mouse, right? Smaller than normal. But whether you get a dwarf mouse as a result of that mutation depends on which parent has passed on that mutation. All right? So let's look at two scenarios here. Scenario number one, we see an offspring inheriting a mutant IGF2 allele from mom mouse, right? From the maternal line. And what we would see under that scenario, if this shaded version of this IGF2 allele is meant to represent the mutant allele, because the maternal allele is methylated and not expressed anyway, that's going to have no impact, right? We still have the normal IGF2 allele being expressed from the paternal chromosome, and so we still get a normal sized mouse. But what happens if the mutation is inherited from the paternal line, if dad passes on the mutation? So in this case, this darker shaded allele is the mutant IGF2 allele being inherited from dad mouse. The normal allele on the maternal chromosome still methylated, always methylated, and therefore silenced, and we end up with a mouse that has no IGF2 expression, right? It has a normal allele, but that normal allele being present on the maternal chromosome has been methylated and silenced, and so this mouse has no IGF2 expression at all, right? Because the version of that gene on the paternal chromosome is the mutant allele. And so this is what's referred to as a parent of origin effect, where in this case, because this is a paternally expressed imprinted gene, right, maternally silenced, we only see the effect of the mutation when it's inherited from the paternal line, because that's the allele that's normally expressed. All right, so this is a really interesting phenomenon, and lots of research going on identifying imprinted genes and trying to understand the significance of this imprinting. But let's talk a little bit about human and how we can see this playing out with a clinical relevance. So there are these two genetic syndromes that have been known about for a really long time: Angelman syndrome and Prader-Willi syndrome. They're very different clinical disorders. If you see somebody who has Angelman syndrome, they will have severe cognitive disabilities, really, really limited language, typically only learning about 5 or 6 words over the course of their life. They have severe seizures that have to be controlled through medication, sort of a constant happy affect with a lot of laughter. So these tend to be very happy children, and very characteristic facial features that are characterized by having a small face, large mouth, and very prominent chin. Prader-Willi syndrome, on the other hand, is characterized by relatively milder disruptions in development. So these individuals tend to have a short stature. They do have characteristic facial features. They have mild to moderate cognitive disabilities and developmental delay. They exhibit reproductive dysfunction. They're typically infertile because of hypogonadism, so their reproductive organs don't develop normally. And they exhibit morbid obesity due to what's called hyperphagia. So these are individuals who have a disruption in the regulation of appetite by the hypothalamus, and as a result they constantly feel like they're starving. And so if behavioral interventions aren't implemented, these folks will actually eat themselves to death. They will just eat and eat and eat until they die of morbid obesity. So it has to be very tightly controlled, but very different clinical presentations for these two phenotypes, or these two genetic syndromes. But interestingly, they're both caused by a deletion on the same chromosome, right? And that's the long arm of chromosome 15, what's called 15q. So here's our chromosome 15, human chromosome. Here's our centromere in the middle, this little constricted area, our two telomeres at the end. The short arm of a chromosome is typically referred to as the P arm. The long arm of the chromosome is referred to as the Q arm. So a deletion on the long arm of chromosome 15 can result in either Angelman syndrome or Prader-Willi syndrome. But what determines which one is exhibited is who the deletion is inherited from. If the deletion is inherited on the maternal chromosome, then we end up with Angelman syndrome. If the deletion is inherited from the paternal chromosome, in other words, we have a normal maternal chromosome but a deleted paternal chromosome, we end up with Prader-Willi being exhibited. And the reason that we have these two very different clinical presentations of genetic syndromes, depending on who the parent was that passed on the deletion, is because this chromosome 15 has a cluster of oppositely imprinted genes. There are about 20 genes on this chromosome 15, maybe a little bit more, that have an opposite imprinting pattern, with some of the genes being expressed from the paternal chromosome and other genes being expressed only from the maternal chromosome. So this is looking at the long arm of chromosome 15, chromosome 15. So here's our centromere, and here's the end of the chromosome telomere, and here's the cluster of imprinted genes highlighted in blue and orange. All of these genes adjacent that are green are not imprinted. They are expressed from both the maternal and paternal alleles. All of these genes that are highlighted in blue are normally expressed from the paternal chromosome and silenced on the maternal chromosome. So blue equals paternally expressed, and orange is the converse. Maternally expressed genes that are normally methylated and silenced on the paternal chromosome, right? And this little segment right here in between these two genes that's labeled or highlighted as IC is what's referred to as an imprinting center. There's a sequence of nucleotides in this region. This is where the methylation pattern is established to either silence this set of paternally expressed genes on the maternal allele, or to silence these genes on the paternal allele and leave them expressed. So you can see control elements in imprinting that are acting at a very distant site to control expression of a gene that's far away in the genome. This is a very common characteristic of imprinted genes. And interestingly, this sequence controls both the methylation pattern that controls the blue genes here and the orange genes, right? Okay, so how does this play out? Well, let's say that this is the paternal allele inherited from dad. This is, or allele, a chromosome, sorry, we're not looking at one gene here. And this is the maternal chromosome inherited from mom. We would see a methylation pattern on these chromosomes such that on the maternal allele, we'd see the methylation mark established here in the imprinting center that's going to silence every one of these blue genes, right? But leaving the sequence that controls our two maternally expressed genes unmethylated and unsilenced. And we would see an opposite imprinting pattern. So in our imprinting center, we'd see a methylation mark established in the sequence that controls our orange genes over here. And so they would actually be knocked out in their expression on the paternal allele. And so in a normal individual who has inherited one copy of chromosome 15 from dad, one copy of chromosome 15 from mom, these genes highlighted in blue would only be expressed in half dose only from the paternally derived chromosome, and these genes highlighted in orange would only be expressed in half dose and only from the maternally derived chromosome. So what would happen if you are an individual and you inherited a chromosome 15 that had a deletion across this region from mom? You inherited it from mom. If you only had this chromosome 15 inherited from dad, you'd be lacking expression of these two normally maternally expressed genes, and you would end up with Angelman syndrome. So the lack of expression of this UBE3A and ATP10A gene caused by a deletion inherited from mom would result in Angelman syndrome. On the other hand, if you inherited a maternal chromosome and inherited a deletion of chromosome 15 from dad, then you'd have the opposite scenario, right? Where you would have only copies of this gene on a chromosome where they're silenced, and so you'd be missing expression of all of these other genes that are marked in blue, and that would give you the phenotypic spectrum that's associated with Prader-Willi syndrome instead. So same region of the chromosome, right, from here to here, but containing oppositely imprinted genes, gives us this very characteristic disparity in clinical presentations for these. If we actually look at what these genes are doing, there's not a lot known about the specifics of what goes on here to cause either Angelman or Prader-Willi, but just to give you a little bit of a sense of what types of genes are here, if you look at the names, these are actually SNRPs. So this SNRP-N, these are also splicing factors. So these are all associated with splicing. This UBE3A is actually a ubiquitin ligase gene, very similar to our anaphase promoting complex that we talked about before. Having said that, even though we know what some of the functions of these genes are, we know very little about how that gene function or lack thereof leads to the problems that we see associated with individuals that have Angelman or Prader-Willi. All right, so the phenomenon of genomic imprinting is not just some esoteric interesting inheritance pattern that's pretty rare. It also has a clinical relevance as well. So just like we saw with mitochondrial inheritance, we see parent of origin effects with this imprinting inheritance pattern, right? When we're thinking about inheritance of a mutation where the allele is only expressed from one chromosome and not the other, then it depends on which parent you inherit that mutation from as to whether you're going to express the phenotype associated with that mutation or not. If the mutant allele is silenced by methylation, you're not going to be expressing that mutation phenotype, whereas if the mutant allele is not silenced by methylation, you will, right? So it's essentially sort of what we see with sex-linked inheritance, where males only have one X chromosome, you don't have a backup. Same thing here, you don't have a backup. So in this pedigree, what we're seeing is inheritance of a mutation where the maternal allele is methylated and silenced. So we see methylation on the maternal allele and silencing, and the paternal allele is not. So we only see expression from the paternal allele. And the shaded individuals are showing the phenotype associated with this mutation. So if we take a look at this inheritance pattern, what we can see, we can pick out two clear indications of a parent of origin effect. If we look at these generations 3 and generations 4 and look at all the affected individuals, we see that in each case those affected individuals have inherited this mutation coming from the paternal line, right? So these individuals have inherited from dad, right? So they're showing the phenotype associated with the mutation, and they've inherited from dad, meaning that the mutant allele is expressed because it came from the paternal lineage. On the other hand, right, this individual clearly has inherited the mutant allele, even though he's not showing it because he's passed it on to his daughter. But who did he inherit the mutant allele from? He inherited it from mom, right? We know he has to have it because he passed it on to his daughter. He had to get it from his mom, who's a member of this family in which this mutation is segregating. But because the maternal allele is typically silenced for this gene, he doesn't show the phenotype associated with the mutation, even though he has that allele. Right? But when he passes the gene on to his daughter, methylation marks are reestablished to match his sex as the new parent, and so that mutant allele is now paternal and is expressed, and we see it showing up in his offspring. So this is what we mean by parent of origin effects, and again, it's all down to the fact that we see in gamete production the silencing of one of the allele according to the new parent of origin that's producing those gametes. So there will be an extra credit question on the genetics problem set that you can attempt to answer if you want. It's extra credit, totally optional, but it has to do with this imprinting and the characteristic inheritance pattern that we see associated with imprinted genes.